production of high energy intermediates. So let us begin. So these are the two major parts of the light reactions. First is photolysis of water and evolution of oxygen, which go hand in hand, right? So we saw how water was split at photosystem 2 and it released oxygen, and the 4 electrons were given back into the photosystem. Clear? And today we will be seeing formation of the high energy intermediates, that is ATP and NADPH. This process, okay, formation of ATP or NADPH, it happens through two pathways. Okay, one is called cyclic uh, photophosphorylation and the other one is called non-cyclic photophosphorylation. Cyclic photophosphorylation requires only one system, which is photosystem 1. It produces only ATP. Okay, there is a transfer of electron from PS1 and it comes back to the PS1, and in this whole process there is production of ATP. However, in non-cyclic ATP synthesis or photophosphorylation, the electron leaves from photosystem 2, it travels through the electron transport chain, reaches photosystem 1 and is finally given to the terminal electron acceptor, which is NADPH. We will see both of these types in detail, so do not worry. Before we can begin, let us just have some basic concepts. Okay, in the electron transport, what really happens is, remember the photosystem. Okay, it is made up of light harvesting complexes plus the reaction center chlorophyll, right? The reaction center. So in the reaction center chlorophyll A, look here, this is the pigment molecule, this is the chlorophyll A. When the photon falls on this pigment molecule, there is rise in its energy. Okay, there is increase in its energy and it becomes excited. That is what you call excited. Okay, it has some extra energy which it got from the photon. What happens to its electrons? Okay, so the electron, which you can see here, it is in the inner orbit. When the photon falls on it, it gets excited and it jumps to the higher energy level. Okay, it jumps to the outer orbital here, and this way, as more and more photon will fall, if let us say one more photon falls, this electron will be released. Okay, it will be released from this atom. Fine. These excited electrons are picked up by the electron acceptor. Okay, so this here is your light harvesting complex, in between where there will be chlorophyll A. Okay, in your photosystem, that chlorophyll A has an excited electron. That excited electron is picked up by some electron acceptor. Clear? These electron acceptors are nothing but they are molecules which can accept one more electron. Fine. They are normally placed closer to this photosystem. Fine. Once they accept the electron, they do not keep it. Okay, they are passed down. So that electron, it will be passed over to the electron transport system. Electron transport system is nothing but it is also made up of different components, most of which can be the type of cytochromes. If you have never heard of cytochromes or you do not know exactly what they are, let me tell you. So remember heme group, this here is the heme group. Okay, the ring here is called porphyrin ring. So you can see this iron in between. Fine. And surrounding it there are nitrogen. Okay, let me just clear it up. There is iron in between, Fe, and surrounding it there will be 4 nitrogen. With these 4 nitrogen, it is forming these rings. Can you see this ring? 1, ring number 2, ring number 3, and ring number 4. Clear? And then it also has this certain tails. Okay, so this structure, this kind of organic molecule is nothing but it is a porphyrin. Okay, it is called a porphyrin ring. So in heme, which is found in your myoglobin, it is found in your hemoglobin, iron is present in this porphyrin ring. Similar to this heme, there are many different types of heme. So this one is heme A. As you can see, there are some differences in this side chain, right? So this heme A is present or can be present in your different cytochromes. Now, coming back to the cytochromes, they have heme group. Okay, I hope you understood what heme groups are. Okay, they are nothing but these kind of porphyrin rings with iron center. So cytochromes are proteins which have this heme group. Fine. These cytochromes, they take part in redox reaction. How do they take part in redox reaction? Because they have this iron, okay, Fe. So this iron, it can go from Fe2 plus to Fe3 plus and Fe3 plus to Fe2 plus. Okay. So it has these two states. The Fe3 plus state, Fe3 plus, will accept electron. Okay, there will be, whenever there is electron which is transferred to it, it will accept an electron and it will become Fe2 plus. Fine. So this is how it will get, what do you call it, reduced, right? When you accept an electron, it is called reduced, being reduced. So this Fe2 plus is the reduced form. Now Fe2 plus will donate its electron to the next transporter, and in this scenario it will become again Fe3 plus and will release the electron and give it to the another molecule. Clear? So this is what happens in cytochromes, and that is why cytochromes, okay, they take part in redox reactions. Remember, they are present in the cytochromes, they are present here in the photosynthesis also, and we shall see them in respiration as well. And also remember some enzymes, okay, like catalases, and some proteins like hemoglobin, myoglobin also have cytochromes, or not cytochromes, sorry, also have this porphyrin ring. Clear? So cytochromes are protein molecules which have this heme group, okay, and they do redox function. Fine. Moving ahead. Now, how is this electron transported from which direction to which? Let us say you are playing passing the parcel. Then, should the parcel be given clockwise direction, anti-clockwise direction? Should you throw it away to the opposite person? How is it supposed to be? So there are some rules, right? So same way here also, that electrons only can be transported in one direction. Okay, so let us see how it is happening. The movement of electrons is downhill. Okay, as you can see, if there is an excited electron which is picked up by some electron acceptor, after that it moves downhill. Okay, it goes down on redox potential scale. Here is this redox potential scale. Let us understand what is redox potential. It is normally written as E red. I don't know if you have studied cell, okay, in chemistry, and you have understood this redox potential properly. Okay, I hope so, but otherwise for our scenario, let us understand redox potential. It is the measure of tendency. Okay, what is it? It is a measure of tendency to acquire or lose electrons. So nothing, these are just numbers. Okay, these are normally numbers in millivolt, microvolt, or volt, depends, but here we are mostly considering millivolts. So these are nothing but they are numbers which show you whether this particular chemical, will it accept electron or will it donate electron. Fine. So just looking at the number, you can come to know, will it be accepting electron or will it be giving electrons. So that is what redox potential is. Fine. Now how do you understand from the numbers? So if it is on positive side, okay, if the redox potential is positive, it means the chemical or that compound has a potential or tendency to accept electron. Fine. These are the chemicals or compounds which accept electron. If they accept electron more, they are said to have positive redox potential. And the opposite is true for negative redox potential. They have tendency to donate electron. Clear? Chalo. Let us see this both things in this diagram. Fine. Can you see here you have this P680, which is PS2. This PS2, with help of sunlight, it is getting excited. This star shows that excitement, okay, that it is in excited state. As you can see, its redox potential, okay, is going from positive, which means it will accept electron, tendency to accept electron, to its tendency is going to negative, which means it is now capable of donating the electron. Clear? So what happened here in between? As you can see, water molecule will give an electron to P680 or PS2, as we shall see later. And this electron, it gets excited, it reaches higher energy level. At higher energy level, it can easily pass on that electron. Fine. So here from negative reduction potential, okay, from negative, sorry, redox potential, minus 900 redox potential, which is as much as it is negative, it is able to donate the electron. So from this reaction center, it will be picked up by some other molecule, okay, which is an electron acceptor. So here this electron acceptor will take that electron. If you see the difference, this PQ, okay, is more positive compared to minus 900, okay. It may be on 0 on redox potential, but in comparison, when you compare, then minus 900 is smaller than 0, right? It is smaller than 0. So what happens is, from more negative, the electron will transfer to positive. Then this cytochrome B6F complex is more positive than PQ. So the electron will transfer to them. From cytochrome B6F, it will transfer to the PC, which is plastocyanin, because its redox potential is even more positive. Fine. So this is how you can see that electrons are transferred only in one direction. This PQ cannot give back its electron to P680, okay, or PS2. Fine. Why it cannot give? Because it is positive compared to the PS2, which means it has tendency to accept electron, but it cannot donate electron to the ones which have negative redox potential. Fine. So this is how the electron transport will occur. Just remember this much. How will you decide, and how is the direction decided? The direction is decided based on redox potential. Clear? The more negative it is, the more it will donate electron. The more positive it is, the more it will accept electron. Fine? And you always have to see in comparison what is happening. Chalo. Now coming back to the original topic, which is formation of high energy intermediates. This is nothing but production of ATP and NADPH. We start with production of ATP, okay, and we shall see in the next. Let us just start with production of ATP, okay. This is called photophosphorylation. Phosphorylation is addition of phosphate. Where is this addition of phosphate, or how is it taking place? It is taking place with help of light, so it is called photophosphorylation. There are three types of phosphorylation, okay. Three types of phosphorylation are: first one, photophosphorylation, which you will see in photosynthesis. Then there are substrate level phosphorylation, which you will see in glycolysis. And then there is oxidative phosphorylation, okay, oxidative phosphorylation, which we shall see after the oxidative respiration, okay. So aerobic respiration we shall see in the next chapter. Fine. In this chapter we are focusing on the photo respiration, which is my mistake, sorry, photophosphorylation, which is nothing but phosphorylation in presence of light. Now photophosphorylation are of two types. It can be cyclic, okay, or it can be non-cyclic. The cyclic photophosphorylation can produce only ATP. Clear? Only ATP will be produced. But non-cyclic photophosphorylation will produce ATP. It will also produce oxygen and NADPH. Clear? So there are two types. There is cyclic and there is non-cyclic. Cyclic produces only ATP, and non-cyclic produces ATP, oxygen and NADPH. Let us start with cyclic photophosphorylation, which is the simplest. What happens in cyclic photophosphorylation? Only PS1, which is photosystem 1, PS700. Okay, all of these can be used for the same thing. So only photosystem 1 here can undergo cyclic photophosphorylation. Let us see what is happening. So here you have PS1, photosystem 1. When there is light which falls on it, it gets excited, and in excited state it can donate 2 electrons. Okay, so it is excited, it is donating 2 electrons. These electrons, they pass through the primary acceptor. Okay, that primary acceptor is nothing but something which will pick up that electron. Once that electron is picked up, it will be given to the next electron transporter, which here is ferredoxin. Okay, ferredoxin is written as FD. Clear? Ferredoxin is a iron rich protein. Fine, it has iron, and as you can see from the name, okay, ferre comes from ferrous and doxin. So this redoxin means it is undergoing redox reaction, okay, reduction and oxidation. And ferre means it has iron. So the name is ferredoxin. Clear? So from the primary electron acceptor, it will pass to the first electron transporter, which is ferredoxin. Ferredoxin will give it to PQ. PQ is plastoquinone. Plastoquinone is a lipid molecule, okay, lipid type of molecule, soluble in lipid, okay, which you will see on the thylakoid membrane. Fine. After plastoquinone comes a cytochrome, which is called cytochrome B6F. Okay, it is just here divided in two parts. So you have cytochrome B6 and cytochrome F. Just remember, I had shown you heme groups, right? So heme A and heme from hemoglobin was shown. Same way, if you have heme B type, then the cytochrome is called cytochrome B6 type. If there is heme F type, then the cytochrome is called cytochrome F. Clear? So these are nothing but these are proteins which have the heme group, okay, which have the porphyrin ring, and they will undergo redox reaction. So the electrons are passed on from plastoquinone to cytochrome B6F. From cytochrome B6F, they are passed on to plastocyanin. Clear? And this plastocyanin is nothing but it is a protein molecule. Okay, it is a protein. This small protein will carry the electron back to the photosystem 1. So can you see, in the whole process, the electron is getting excited from photosystem 1. It is passing through different electron transporters and it is coming back to the chlorophyll 1. Okay, hence it is called cyclic photophosphorylation. As you can see, it is moving in a cyclic manner. The electrons which are leaving photosystem 1 are coming back to photosystem 1. During this whole process, there is production of ATP, which we shall see later on during this lecture. Okay, in the end of the lecture, we will see how ATP is produced. But just for your information right now, know that when these electrons are getting transferred through electron transport chain, that will cause formation of ATP. Hence, it is called cyclic because the whole thing is cyclic photophosphorylation. Okay, in presence of light, ATP is generated. Now let us see non-cyclic photophosphorylation. Cyclic photophosphorylation included only photosystem 1. Non-cyclic includes photosystem 2 and photosystem 1. Here it starts with photosystem 2. Okay, the starting point is photosystem 2. Okay. Now there is light falling on photosystem 2, which is of 680 nanometer. Fine. The 680 nanometer light will be absorbed by the chlorophyll A of the reaction center, okay, of photosystem 2. It will get excited and it will donate its electron to the primary electron acceptor. Fine. Now once it donates its electron, okay, from PS2, photosystem 2, the chlorophyll has given out one electron. That chlorophyll will now be lacking one electron, right? There will be some deficiency of electron. How is this deficiency what you can say quenched, okay, or fulfilled? So remember the photolysis of water that comes in handy here. Even during photolysis, we had discussed it happens only at photosystem 2. So let us see what is happening. Two water molecules in presence of light near photosystem 2, specifically at the oxygen evolving complex, in presence of manganese, chlorine and calcium, will get converted into 4 electrons, 4 protons and 2 oxygen molecules. Um, O2, okay, 2 oxygen, sorry, 1 oxygen molecule and 2 oxygen atoms, fine? Okay, so during this process, which is photolysis of water, the oxygen will be released now. The H plus, we will see what happens to it later on. And the electrons, okay, the electrons, they are given to the photosystem 2. Fine. So whatever electrons photosystem 2 lost because it gave it to the electron transporter are gained back from water. Clear? So here water is acting as electron donor. Water is giving electrons to photosystem 2. This photosystem 2 is getting excited and giving electrons to the primary electron acceptor, which is sometimes referred to as pheophytin. Okay, it is the primary electron acceptor. It will accept the electron. Remember this primary electron acceptor and the chlorophyll A molecule, they form together the reaction center. Fine. Now from this primary electron acceptor, the electron will move to the next electron transporter, which is called plastoquinone. I already told you quinone are nothing but they are lipid soluble molecules. Okay, if you find it in plants, you call them plastoquinone. Okay, what they will do is they will pick up these electrons from the electron acceptor and they will move it ahead. Okay, they will give this electron ahead to cytochrome B6F complex. Now from this cytochrome B6F complex, you will give the electrons to plastocyanin. Clear? So can you see this is the whole electron transport? Fine. From PS1 to pheophytin or primary electron acceptor, from primary electron acceptor to plastoquinone PQ, from PQ to cytochrome B6F, from cytochrome B6F plastocyanin, and now this plastocyanin will give the electrons to PS1, okay, photosystem 1. Now the reaction center chlorophyll of photosystem 1 will get excited when the light of 700 nanometer falls on it. As it gets excited, it can donate electrons, so it will donate electron to ferredoxin. Okay, remember this is the same protein that we studied in cyclic photophosphorylation. So it will give its electron to ferredoxin. Ferredoxin will give the electrons to an enzyme called NADP plus reductase. Clear? NADP plus reductase. Just recall the chapter which we studied previously, which is biomolecules, okay, in which we discussed enzymes. So in enzymes, remember the first class of enzymes was oxidoreductase. These oxidoreductases have dehydrogenase, reductase, all this kind of enzymes which do the function of redox reaction, right? So here what is happening is this enzyme reductase, when you have A's in the end, you know it is an enzyme. What is it doing? It is reducing NADP plus, hence the name is NADP plus reductase. Clear? So ferredoxin will give its electron to the enzyme, and this is the reaction that will happen. NADP plus 2H plus, okay, which we are getting from here. Photolysis of water, those two hydrogen ions or two protons, 2H plus, will be added to the NADP plus and give you NADPH. Now normally there is just this normal NADPH, but scientists have found that it is associated with one more proton. Okay, so this one more proton is normally connected to the NADPH molecule without any covalent bond. Okay, it is just present there. So this here is the NADPH. And this is how you are making ATP during electron transport, and you are making NADPH finally. If you remember, in respiration, the terminal electron acceptor, okay, it is a molecule which will accept the electrons at the final position, is oxygen. In this case, the terminal electron acceptor is NADP. Okay, so this NADP is terminal electron acceptor. As soon as it accepts that electron and two protons, it turns into NADPH, reduced form of nicotinamide adenine dinucleotide. Okay, so in this whole process of non-cyclic photophosphorylation, you are making ATP during electron transfer as well as making NADPH as the end reaction. Fine. Will you be able to remember this whole process? Quite simple, right? You start with PS2. PS2, when it gets excited, it donates its electron. That electron are filled up from the splitting of water, okay? From photolysis of water, the electrons get replaced in photosystem 2. Fine. These electrons which left photosystem 2, they are accepted by the primary electron acceptor. They pass through plastoquinone, cytochrome B6F, plastocyanin, and given back to photosystem 1. These electrons from photosystem 1, they get excited again. They are passed to ferredoxin. Ferredoxin gives them to NADP reductase, which is an enzyme that converts NADP into NADPH. Clear? So an analogy for remembering this process, okay, analogy is some example, okay, some similar example from which you can understand this or remember this is, think of this construction site, okay. So here a worker, he is providing all the energy and all the force to in form of photon, okay. What is this photon doing? It is exciting that electron, so that electron is leaving. So if you put a hammer on this seesaw, the electron, it will jump, right? So here the electron is jumping. It is caught by photosystem 2. Clear? Now this photosystem 2, when it is in excited state, it will donate its electron. This electron passes through this electron transport chain. Okay? Electron transport chain is the reason why you are able to produce ATP. As the electron, it loses energy, it comes back to this position, okay, in photosystem 1. Now photosystem 1, another photon, it provides energy and that electron jumps again. This electron is caught up by your NADP reductase and it will provide the electron to NADP, forming NADPH. Clear? So I hope it will be easier for you to remember. A very detailed version of the same thing, how it is happening, just for your information, okay, how you will be able to see it on a chloroplast, um, this thing, thylakoid membrane, okay, how you will be able to see it in thylakoid membrane. This here is the chloroplast stroma, and this here is the lumen. So all of these processes are taking place in the lumen, okay, of the thylakoid. Remember, light reaction occur in the membranous system of chloroplast, right? Fine. We started with PS2. There is light falling on it. So the electron, they get excited, they are passed on. These electrons which are leaving PS2 replaced by photolysis of water. Electrons passed on to PQ, which is plastoquinone. As plastoquinone, it accepts an electron. It also accepts protons, okay, so it becomes PQ to PQH2, reduced plastoquinone. Reduced plastoquinone, it is, as you can see, taking up H plus from outside, okay, from the chloroplast stroma. It will give this electron to the next, which is cytochrome B6F. As it donates its electron, it also releases the protons, okay, and becomes PQ again, oxidized state of plastoquinone. Now this electron from cytochrome B6F is passed on to plastocyanin. Clear? Plastocyanin is a protein molecule. It will transfer it is mobile, okay, it can move around. It will take electrons from cytochrome B6F and give it to photosystem 1. Photosystem 1, again, when it gets excited, it will donate its electron to ferredoxin. Ferredoxin will give it to ferredoxin NADP reductase. and which will transfer the electron finally to NADP plus, forming NADPH. Clear? So this is how the whole reaction is taking place. In case of cyclic photophosphorylation, just remember what was happening. You had photosystem 1, the electron got excited, it was passed on to ferredoxin. From ferredoxin, it was going to, where was it going? It was going to the plastoquinone, right? Plastoquinone, plastocyanin, and coming back to the photosystem 1. Clear? So this reaction is happening here. Instead of ferredoxin passing that electron to the NADP reductase, it will pass its electron to the other electron acceptor. Clear? And that is how the cycle was cyclic. Chalo. This transfer of electrons starting from PS2, this is PS2, it started from PS2 and to PS1 to NADPH final, is called Z scheme, okay, because it forms this Z-like structure. I don't know, it looks more like N, but when you look at it from this side, okay, it looks like a Z. So it is known as Z scheme. Remember, this is an important point. Non-cyclic photophosphorylation looks like a Z scheme. Fine. Now we saw everything which was simple. Now let us talk about how the ATP is actually synthesized. Okay, at what point or at what phase will it be synthesized properly? So this can be explained by chemiosmotic hypothesis. Clear? If you have understood the phosphorylation properly, then only you will be able to easily understand this process. Right. So ATP synthesis is linked to development of proton gradient. Okay, gradient is what? On one side, on one end, some concentration is higher compared to the other side, right? So that is called gradient. What is this gradient made up of? It is made up of protons. Fine. And where is it present? So it is present across the thylakoid membrane. Fine. How will you see this gradient? So there will be more proton towards inside of the thylakoid. So there are too many H plus ions inside the thylakoid and less H plus ions in the stroma of chloroplast. Clear? Comparatively, there is less protons in the stroma, more protons in this lumen. There is proton accumulation in the lumen because of, there are some reasons why there is H plus higher inside this thylakoid compared to outside. Let us see these reasons one by one. It is just sort of revision. Remember that splitting of water takes place on the inner membrane of thylakoid. Okay, so inside this thylakoid only splitting of water is taking place. So here you can see H2O. It is splitting, it is giving off electrons, it is giving out oxygen and protons. So because of this, the proton concentration inside the lumen is high. Second reason, as the electrons are transported, okay, through this electron transport chain, as we just saw that plastoquinone, it picks up the proton from stroma, okay, from outside thylakoid, because it cannot just transfer electron, it has to get reduced, okay. So it will take electron plus proton, then it will become PQH2, which is reduced plastoquinone. Reduced plastoquinone will give its electron to the next electron transporter, which is cytochrome, and it will release the H plus in the thylakoid lumen. Okay, so it is releasing more and more H plus in the thylakoid, which are picked up from outside. So because of this also there is higher H plus in the thylakoid lumen. Also, the cytochrome B6F also has been shown to transfer some amount of protons. Okay, it also transfers some amounts of proton towards the thylakoid lumen. Fine. And finally, the NDP reductase, the final enzyme of non-cyclic photophosphorylation, is located towards stroma. So if you see this, photosystem 1, 2, plastocyanin, photosystem 2, photosystem 1, plastocyanin, and near the photosystem 1 you have ferredoxin, FD, then you have this FNR, which is nothing but your NADP reductase. Okay, NADP reductase, if you see, It is present outside the thylakoid membrane, okay, towards the stroma. So it is present near the stroma. It will convert NADP to NADPH. During this process, it is utilizing one more H plus. Okay, it is using one more H plus from stroma. So basically, what are you doing? You are reducing the H plus from stroma and you are increasing the H plus inside the thylakoid lumen. Clear? This is what is happening. Fine. So now you have more H plus here in the lumen. What will happen to this H plus? So according to the we will see that in transport, but any chemical or any compound, it moves from its higher concentration to low concentration. So here there is this high electrochemical potential gradient. It is higher inside and it is lower outside. So these protons will of course try to move outside, clear, from their higher concentration to their lower concentration. When this movement happens, when these protons they move from inside to outside, at this point ATP is synthesized. The molecule or the protein which converts this energy into synthesis of ATP is called ATP synthase. Okay, this breakdown of gradient, okay, from high concentration to low concentration when protons move, that is the process which synthesizes ATP. This gradient is broken down by ATPase, okay, or ATP synthase. Let us see the structure of this enzyme. This here is the stroma part, okay. Stroma means outside the thylakoid membrane, okay. So this here is the thylakoid membrane, lipid bilayer, okay, and this is the inside portion, which is the thylakoid lumen. The lumen has higher H plus concentration, right, and outside there is low H plus concentration. This enzyme is made up of two parts. It is made up of F0 here and F1 here. So this is F0 and this is F1. Okay, these are the two particles. Normally it looks like a lollipop shaped, okay, structure. Here it will be bulging and this is its what you can say fixed part in the lipid bilayer. Okay, so F0 is always fixed inside the lipid bilayer while F1 is protruding out. Now what will happen? So when H plus it enters from the thylakoid lumen, because remember thylakoid lumen has higher concentration of H plus. This H plus, there is a sort of channel, okay, which is shown here, a straw like structure. Through this straw the H plus will go and bind here. When it binds, now this whole thing, okay, this is cycle like structure, okay, it will move. I always give an example. If you have gone to the gardens, okay, in the gardens they have this sort of structure through which you have to enter the garden. If this is the garden, let us say wall, and in between you have this it will be this sort of thing, okay, fine. So if you have to go, you have to stand here and then you have to push and this whole thing will rotate and then only you can leave on the other side. I hope you are able to understand. When you want to enter a garden, there are these, I don't know what they're called, okay, at the gates you find these kind of cylindrical structures, okay. You enter from one side and then you have to turn and then only you can enter into the garden, okay. Chakedi or something it may be called, I don't know. So same kind of thing happens here. The H plus it joins, it has to rotate this whole thing, okay. As soon as it joins, the whole F0 particle, it moves, it rotates. The direction is shown here. As it rotates, it will move like this, and now this H plus will be here, okay. After whole movement, okay, after whole circle, the H plus comes here. At this point it is free to leave. Now this H plus will be able to leave and enter the stroma. So this is how this H plus is moving the rotor, okay, it is moving this rotor. Now as this rotor moves, it is connected to this F1 part. So the F1 part also moves. As you can see, it is made up of alpha and beta, and there is also delta which is connecting these two again. So when this F1 particle moves, during its movement, there will be enzyme catalysis and it will produce ATP from ADP and inorganic phosphate. This is how ATP is synthesized. Clear? Remember, ATP and NADPH are required for the next phase of photosynthesis, so they are synthesized in the stroma. ATP also is synthesized in stroma, and we saw that the NADPH is also synthesized in stroma. Clear? Fine. See you next time. We will start with the dark reactions, okay?